Thank you. I would like to thank the organizers for their very kind invitation. Work in my lab focuses on neurovascular mechanisms of inflammation and tissue repair. And today we'll be discussing exciting new findings for the role of blood proteins as drivers for neurodegeneration, and share our perspective how to harness this information for the development of new therapies for Alzheimer's disease. These are my disclosures, funding for the lab. I'm a founder and chair of the Scientific Advisory Board of a university spinout, and also named inventor on patents and patent applications, some of which are related to the presentation today. A common thread among neurological diseases with diverse etiologies is leak of blood proteins into the brain. And this is also the case in Alzheimer's disease. And here you can see three-dimensional immunolabeling of the blood protein fibrinogen in a cleared brain from a patient with Alzheimer's disease surrounding damaged blood vessels. And these are early events, cerebrovascular alterations, early events in the pathology that also correlate with early onset and worse progression in the disease. And the driving question in our research has been, are these blood protein leaks in the brain drivers causal to neurodegeneration? And indeed, fibrinogen is a protein that is not present in the normal brain. It's produced by the liver. And there has to be some leak of the blood-brain barrier to find access into the brain parenchyma. And here you can see that in a non-demented control, there is no fibrin in the brain. But in Alzheimer's disease, there is abundant presence of this fibrinogen protein in the parenchyma. And in AD, fibrinogen can also bind to amyloid, and there it correlates also with activated microglia. Studies have also found fibrinogen in CAA, where it correlates with severity of CAA pathology, and is also increased in the individuals, Alzheimer's patients with the APOE4 polymorphism. And fibrinogen is also detected in ARIA after amyloid-lowering therapies. In addition to neuropathology, biomarker studies have proposed CSF and plasma fibrinogen as a biomarker for early detection of disease pathology, as well as also progression in Alzheimer's disease. And in these studies, fibrinogen in the fluid also correlates with other AD biomarkers like phospho-tau, A-beta-42, and soluble PDGF receptor beta. Fibrinogen has a unique structure function, is a soluble protein in the blood that is converted to insoluble fibrin at sites of lesion in the brain, and is also a druggable target. So here I have highlighted in the fibrinogen gamma chain the 40411 coagulation domain of fibrin that binds to the platelet receptor. This is how our blood clots. And this is a druggable interaction. There is an antibody called RioPro that is disrupting the interaction of fibrinogen with platelets. And this is manufactured by Eli Lilly and is prescribed as a thrombolytic. So what I noticed is that fibrinogen has another domain, an inflammatory domain, the 377-395 binding site, that is the binding site to the CD11BI domain of complement receptor, which of course, as we know, is expressed on microglia and macrophages and is a key driver for inflammation. So a driving question in our research was, is it possible that when fibrinogen entered the brain, it could be hijacking these receptors on microglia cells? And since these two domains are non-overlapping, could we differentially target selectively the inflammatory domain of fibrin without affecting its beneficial effects in normal hemostasis? So our hypothesis was that blood leaks in the brain are drivers of neurodegeneration, and the experiment was to perform genetic loss of function studies to find if they're necessary for disease. And indeed, the fibrinogen knockout mice, which have all blood proteins except fibrinogen, were tested first and were protected in models of Alzheimer's disease, as well as also other neurologic diseases like multiple sclerosis, parasite-deficient mice, and protection in remyelination. In 2007, we introduced a new genetic tool, the fibrinogen knock-in mouse, that had only a mutation within the inflammatory domain, and this was also protected in Alzheimer's mice, and we showed that this domain was critical for activation of microglia. Targeting the inflammatory domain became the basis for therapeutic intervention by the development of fibrin-targeting immunotherapy, a first-in-class antibody to neutralize toxicity of fibrin in the brain. A first-in-human trial with a humanized version of this antibody started 2 months ago in a safety study, phase 1 safety study in healthy volunteers. To dissect the role of fibrin in Alzheimer's disease, we took a multi-pronged approach where we combined longitudinal in vivo to photon imaging, development of new protocols to detect fibrin and neurovascular pathology in mouse and human brain, as well as also cognitive behavioral tests. What we found, we did endogenous labeling of fibrinogen and amyloid plaques, and this is the 5-XFAD animal model of Alzheimer's disease. And what we found that similar to amyloid, fibrin was also a site of synapse loss in Alzheimer's mice. And here you can see spine turnover with a significant reduction of synapses around fibrin deposits in the brain, showing that this was a new culprit for synapse loss in AD mice. When we crossed the 5-XFAD mice with the fibrinogen mutant mice, there was an increase in the number of neurons and synapses, decrease of LAMP1, a lysosomal marker of degeneration, as well as also decrease in microglia. And in behavioral tests, by superimposing the fibrinogen mutation, we could rescue the defects of 5-XFAD mice in learning and memory, as well as also hyperactivity. In collaboration with Jorge Palop at the Gladstone Institutes, we employed the machine learning approaches for unbiased interrogation of behavioral alterations in AD mice. And in this test, the Palop lab has identified multiple behavioral motifs that change in Alzheimer's mice. And when we superimpose the fibrinogen mutation, these were either significantly reduced or rescued in the absence of the inflammatory domain of fibrinogen in mice. Overall, these results identified fibrinogen as a new culprit of spinal elimination and cognitive decline at sites of vascular damage, showed that microglia activation through this pathway was really critical for dendritic spinal elimination and cognitive impairment. And these studies also provided a new molecular mechanism to explain emerging findings from clinical data that shows that cerebrovascular pathology is an early and independent mechanism for neurodegeneration and cognitive impairment. And as an independent mechanism, these results have also suggested that it might be to also independently targeted to achieve maximum therapeutic benefits in Alzheimer's disease. So with that in mind, we decided to develop a pharmacologic tool, a first-in-class fibrin-targeting antibody to neutralize blood toxicity in neurologic diseases. And of course, the challenge we had to overcome was that this was the most important hemostatic factor, and we did not want to interfere with its beneficial coagulation functions. So for this reason, we developed an epitope-specific antibody that specifically recognizes only the inflammatory domain of fibrin. This antibody can only recognize pathogenic fibrin in the brain. It does not interact with soluble fibrinogen in the bloodstream, and it doesn't have any anticoagulant effects. The antibody showed efficacy in protecting from neurodegeneration and suppressing inflammatory pathways in microglia that are associated with neurodegeneration in human AD in reversal trials after therapeutic administration in Alzheimer's models. This antibody has now been humanized, affinity matured, GMP manufactured. It has completed safety and toxicology studies in 2 species, and the safety trial in healthy volunteers is currently ongoing. 3 years ago was the 20th anniversary of Nature Immunology. It also coincided with the 100th approval of a biologic by the FDA. And Nature Immunology featured 20 antibodies for the 20 years of Nature Immunology, one of which was also the fibrin immunotherapy. And there is where we got an opportunity to discuss how our curiosity about how the blood affects the brain led to the discovery of the first tool to be able to neutralize this pathogenic pathway with implications in across the board of many neurodegenerative diseases that are characterized by neuroinflammation and synaptic dysfunction. These studies also opened new questions about the molecular interface that blood proteins utilize to interact with innate immune cells. And we wanted to address what is the specificity among blood proteins and what are the pathways that they use to activate innate immune responses. And to address this question, we employed a multi-omic pipeline that consists of both transcriptomic and phosphoproteomic studies, as well as also genetic loss of function studies in mice, to be able to define the blood-induced microglia functions in neurodegeneration. We reported the blood microglia network, which is the transcriptomic changes in microglia upon exposure to a wild-type blood, and these are associated with several genes that are involved in neurodegeneration. And surprisingly, when we superimposed on this network the transcriptome of microglia exposed to blood from fibrinogen knockout mice, 97% of these genes were downregulated, suggesting that fibrinogen is a key protein in the blood that induces activation of microglia. And to test if these effects were receptor-mediated, we used plasma from the fibrinogen knock-in mouse that has a coagulable fibrinogen but cannot activate immune functions in microglia. And indeed, this was significantly different, the transcriptome of microglia than microglia exposed to wild-type blood. About 300 genes were downregulated. And we used as a control albumin knockout plasma, where you can see it was rather similar to wild-type plasma, suggesting that blood proteins have a distinct specificity to induce receptor-mediated transcriptional programs in microglia cells. And these results are also in agreement with our prior studies that it's not only microglia activation, but also downstream effects from this pathway that include demyelination and dendrite loss that are also specific through fibrinogen activation. And you can see here that knockout plasma from fibrinogen knockout mice or knock-in mice does not induce these pathogenic effects. To test if this pathway is necessary for neurodegeneration in Alzheimer's mice, we performed single-cell RNA-seq sequencing from microglia isolated from either 5XFAD mice or 5XFAD mice crossed to the fibrinogen mutant mouse. And what you can see here that by only depleting this mutating this immunologic domain of fibrin, there is a reduction in the neurodegenerative signature of the microglia that includes many neurodegenerative genes, as well as also oxidative stress genes, while there is restoration of the homeostatic signature of microglia, showing that in this animal model of AD, fibrin CD11B signaling is necessary for the induction of microglia neurodegenerative genes in Alzheimer's mice. These studies changed the way we thought about the blood-brain barrier, not as a secondary consequence of inflammation, but instead as an upstream driver of pathogenic polarization of innate immunity. These studies also supported that the blood proteins are not interchangeable in their roles to activate the immune response, but instead they can induce distinct and receptor-mediated transcriptional changes in microglia cells. They also showed that fibrinogen is unique among blood proteins through its conversion to fibrin to induce this, to be necessary and sufficient to induce these neurotoxic signatures in microglia cells. And these studies also provided support that neutralizing this pathway could overcome the pathogenic effects of cerebrovascular pathology in the neurotoxic activation of innate immunity. So in the context of Alzheimer's disease, neutralizing blood toxicity by fibrin immunotherapy could be a selective way to selectively inhibit the neurotoxic microglia. And selective inhibition is really important. These cells have many protective functions, including removal of amyloid. And by interfering with the fibrin pathway, amyloid phagocytosis is not affected. So this has increased selectivity than other immunosuppressive therapies. At the same time, this could also protect from synaptic dysfunction that can lead to cognitive protection, especially in APOE4 Alzheimer's patients that have increased blood-brain barrier disruption. This type of therapy can be used alone, but of course also in combination with amyloid-lowering therapies to either increase the efficacy of amyloid therapies in protection from cognitive impairment, but also potentially decrease their side effects by decreasing ARIA pathology. As this pathway is moving towards the clinic, it's really important to also develop tools for sensitive imaging of cerebrovascular pathology, both in neuropathology studies, but also non-invasive tools. And this is a protocol we published in collaboration with Bill Seeley in the Alzheimer's book in January for three-dimensional volume imaging that is essential to detect in a sensitive manner blood leaks in the brain in Alzheimer's disease. I would like to thank the very talented people in my lab who have performed this work, our amazing collaborators, and for the purpose of this talk, Leonard Tmuki, Jorge Palop, Bill Seeley, Fanny Elahi for their support with these studies, and all our funding that has supported our work over the years. Thank you very much for your attention.
Thank you for a fabulous talk. That was really brainstorming. Okay. The second talk we have is actually a virtual pre-recorded talk by Professor Henniker entitled Overview from Basic Science Through to Druggable Target Identification.
Everybody. It's a pleasure to welcome you to this session on neurodegeneration and inflammation. I will give you an overview from basic science through to druggable target identification. My name is Michael Henniker, and I'm working at the Luxembourg Center for Systems Biomedicine. So why do we have this inflammatory reaction in the AD patient brain? Coming from a pandemic, it has been made clear to us by nature that we're constantly exposed to viruses, bacteria, and fungi. And so the major reaction to these agents and pathogens is the innate immune reaction. Now, what is the innate immune system doing for us? This is shown by this little movie where a neutrophil is hunting for a bacteria, in this case, the Staphylococcus aureus. You see that he follows up on it, takes a lot of clever decisions, does not attack surrounding cells until he finally can corner the bacteria, take it up and digest it. And this shows you that the innate immune system is nothing else than our first line of defense. These cells obviously do not only exist in our bloodstream, they also exist in parenchymal organs. In the brain, we call them microglia, alveolar macrophages in the lungs, Kappa cells in the liver, mesangium cells in the kidney, Paneth cells in the gut, and in the bones, the bone marrow macrophages. Now, all these cells protect these organs from infection, but they also contribute to their structure and function. And if you're interested in the history of microglia, these have been identified by Pio del Rio Ortega about 100 years ago, and there's a very elegant overview and review by Amanda Sierra published in Glia some years ago. Now, why do these cells react to beta sheet structured amyloids depositing in our brain? This is showing you a Congo red stained AD patient brain that also shows stain for MHC class 2 positive microglia cells. Now, before Congo red was actually used to stain human brains, it has been licensed by Paul Bötticher 1880 as a bacterial stain, raising the question, what is the commonality between a human brain and the bacterial surface? Now, it took about more than 100 years until Matthew Chapman, in a very elegant paper, found that various bacteria common to us, including Escherichia coli, Staph aureus, Salmonella typhimurium, carry beta sheet-structured amyloids on their surface. And they're pretty similar to those beta sheet-structured amyloids depositing in our brain. In other words, that means that myeloid cells, including these microglial cells, by nature, over 1 million of years of evolution, has been taught that a beta sheet-structured amyloid is nothing else than a pathogen surface. And nature has equipped these cells with so-called pattern recognition receptors, including the toll-like receptor family and others, to sense these structures. And ligation of these toll-like receptors or other pattern recognition receptors starts off a massive inflammatory reaction, characterized by the release of inflammatory mediators. At the same time, these cells stop doing a lot of senseful things in our brain. They reduce their synaptic scaling and pruning, they reduce their production of neurotrophic factors, and they're also much less capable of clearing debris from the brain. Now, this inflammatory reaction can be visualized by PET ligands in patients. This is an example of PK195, TSPO ligand, which, as you easily can see, labels in the AD patient brain the limbic system and neocortical structures, while the age-matched non-demented control case shows only vascular enhancement. Now, there are more danger-associated molecular patterns that accumulate in the brains of AD patients, such as chromogranin A, which is elevated in a number of neurodegenerative disease. And as you can see, here is as strong in inducing an inflammatory reaction by microglia cells as bacterial lipopolysaccharide. Another danger-associated molecular pattern is MRP14, belonging to the S100 protein family. And knocking S100 protein family members such as MRP14 out in the APP-PS1 mouse model lowered TNF-alpha and interferon levels in the brain significantly. At the same time, it enabled microglia cells to take up and degrade amyloid beta much better. Now, there are functional and structural consequences of innate immune activation in the brain. A couple of cytokines we know of, in this case interleukin-1 beta, are capable of suppressing long-term potentiation in the hippocampus, which is an important mechanism of information storage and consolidation, as you know. There are other immune factors, including TNF-alpha, nitric oxide, L6, complement factor 3, and C1q, which have a similar effect. So in concert, these immune factors may substantially contribute to functional impairments in these circuits by suppressing their function. Now, on a structural perspective, there is evidence that microglia cells that do not express so-called do not eat me signals can take up living neurons. This is an experiment from Guy Brown's laboratory showing you that when he co-cultured microglia with neurons, 30% of neurons got lost, and they didn't die by apoptosis or necrosis. They were taken up by microglia cells in an MFG-E8 dependent mechanism. Now, beyond that, microglia cells that are activated by oligomeric amyloid beta have been shown to take up synaptic structures when they're labeled with complement factor 3 in excess. And this is an experiment published by So-Yeon Hong some years ago showing that microglia cells take up in a complement receptor 3 dependent fashion these synaptic structures in an APP-PS1 transgenic mouse model. Now, when we consider that amyloid beta may deposit as early as in the 4th or 5th decade of life, we have to consider that all the mechanisms after we're investigating happen to be present in the coexistence with inflammation. So all the mechanisms we are interested in studying neurodegeneration have to be studied in the presence of inflammation. and inflammatory mediators. Now, inflammation in the brain is being influenced by a number of lifestyle factors, collectively called the exposome. We know that midlife, obesity, poor oral health, aging itself by generating a senescence-associated secretory profile, exposure to infections or systemic inflammation, or reduced physical activity can increase systemic presence of inflammatory mediators, which then may act to aggravate and accelerate disease processes linked to Alzheimer's disease. Brain trauma, another risk factor, is a direct impact on the brain. Now, beyond these lifestyle factors, there are factors we cannot influence, which is our genomic background. And the past decade has experienced the identification of a huge number of risk polymorphisms, of which the majority directly influence innate immune signaling pathways. Now, this is more recent work by Kozo and colleagues published in August last year, which I want to point to by combining attack sequencing and transcriptional footprinting in more than 150 isolates of microglia from AD cases, and analyzing GWAS risk polymorphisms in conjunction with this. These authors found out that while in Parkinson's disease there is no major role for GWAS in microglia cells, in Alzheimer's disease, in strong contrast, GWAS hits may play their major role through modulating microglia activities. Now, this is an example of the human microglia core signature identified by Eric Bodeker's group and published in Nature Neuroscience in 2017. And there you will find a lot of potentially druggable targets. Within those, the so-called inflammasomes, and the inflammasome products, interleukin-1 beta and IL-18. And we have been spearheading the investigations in the Alzheimer context, identifying the NALB3 inflammasome as the major inflammasome present in the AD patient brain, expressed in microglia cells. We also found that acid specks released by dying microglia would seed amyloid beta deposition. And this led us to a patent which targets anti-acid-directed therapeutics in a number of neurodegenerative disease. It also led to collaborations where we searched in over 100,000 plasma samples for anti-acid antibodies in humans, and to the development of blood-brain barrier penetrant NALB inflammasome inhibitor. This cascade of translational steps into the clinical direction can be performed with many of the targets I just showed you in the human microglia core signature. Now, last but not least, I want to point to a role of the peripheral immune system and stimulation. There is evidence that systemic inflammation drives A-beta pathology and also tau pathology. There is further evidence that peripheral cells, such as macrophages or neutrophils, can cross the blood-brain barrier under certain circumstances and cause damage in AD patient brain. Chronic airway-induced inflammation or other peripheral allergies may cause an increase in brain inflammation. And more recently, it has been shown that T cells may drive microglial inflammation and through tau pathology. Now, in summary, I hope I could show you that innate immunity is a driving force behind the initiation of AD pathogenesis, progression, and phenotype development. Lifestyle factors in our genetic background influence neurodegeneration.
大家好，我是侯志坚。
Central immune responses contribute to neural dysfunction, neuronal autosomal apoptosis and synaptic dysfunction. And how inflammatory activation and subsequent microglial apoptosis cause ABL seeding and tau phosphorylation. And systemic inflammatory responses, including allergic reactions, may cause delayed synapse loss, microglial ABL deficit, and tau pathology. With this, I thank you for your attention, and I hope you have many questions. Thank you.
Well, you have two fantastic talks. Okay, we are not taking questions right now. The questions will be in the end. Thank you for understanding. Well, we have two fantastic talks in neuroinflammation, Alzheimer's disease, and I think now we're going to be a little bit more broad, and we're going to talk about drug development in the area of anti-amyloid treatment by Michael Izari from Wilsine.
Thank you so much to the organizers for inviting me to present at this wonderful session. It's really an exciting time in Alzheimer's disease research with the advancements in therapeutics, the refinement of biomarkers, and a greater understanding of the pathophysiology of disease. It really is an opportunity to think carefully about the adaptation of drug development in the era of anti-amyloid therapies. So as we hear about some of the inflammatory targets in this session, thinking more broadly how we would develop those therapies, how we think of clinical trials at a time where there may be available approved anti-amyloid therapies, and how to conceptualize the patient population and the clinical trial design. I'm an employee of AZI. And with the approval of anti-amyloid therapies in the United States and the opportunity for changes in the therapeutic landscape, there are several clinical trial considerations that can be brought up in terms of how to select patients, how to conduct clinical trials, how to think about combination therapies. So, for instance, anti-amyloid therapies are not yet established standard of care. So in thinking of a clinical trial of a new therapeutic in Alzheimer's disease, and in particularly early Alzheimer's disease, the question can come up whether or not to allow amyloid therapies in that clinical trial. Certainly by only including patients that have never been exposed to amyloid therapies, you may have a more homogeneous population in order to detect a treatment effect in the new target. However, that could impact recruitment or enrollment if a significant proportion of people start using anti-amyloid therapies. There is also the question of whether anti-amyloid therapies are prohibited during the course of an Alzheimer's clinical trial, which can range for 18 months or longer. If the decision is to allow anti-amyloid therapies in a clinical trial for a new target, then how to implement that, how to operationalize that. One option would be just normal clinical care, whatever the patient comes in at, whether they're on an anti-amyloid therapy or not, whether they've been exposed to one or not, just take them and then randomize them to the new therapeutic. Again, that could add heterogeneity depending on the outcome and the particular target. It might simplify it to select a single anti-amyloid therapy, perhaps a backbone anti-amyloid therapy. That's one drug that allows standardization in terms of the titration, the safety of a particular molecule, and the rate of amyloid clearance. Alternatively, multiple anti-amyloid therapies can be allowed, but you may want to standardize to the amount of amyloid that's been achieved. So you may want to focus on patients that have been on chronic anti-amyloid therapy and have amyloid cleared to below what's considered amyloid negative. The other consideration then is the study population. I'll talk about this in more detail in terms of a biomarker-defined study population. But a clinical trial in early AD will have to consider potentially approved therapeutics that are already available for early AD. While in preclinical AD, it may be easier to run a clinical trial that's not on top of anti-amyloid therapeutics. And then a biomarker-defined population may better define the timing of treatments and depending on the target of the new therapeutic. If an anti-amyloid therapy is combined with a new therapeutic, then there's a range of considerations in terms of clinical trial designs. Full factorial design that includes a placebo that may be on neither treatment, as well as the monotherapies and the combination therapy, or an adjunctive design where a treatment or placebo is added on top of a backbone of kind of open amyloid therapeutic. Considerations of dosing sequences. You may want to treat with an amyloid therapeutic first and then a tau therapeutic or a neurodegeneration therapeutic. There are considerations in terms of the different dosing modalities, whether it's an ASO, small molecule, or monoclonal antibody. And if you're thinking of combinatorial designs, if you're including dose ranging studies within that, then that actually makes it even potentially more complicated. An important factor of the outcomes in these clinical trials is really demonstrating the impact of each therapy on the disease, and that can be both on biomarkers and clinical outcomes. And then also understanding the impact of safety of each of the therapeutics and how the safety issues may overlap depending on the treatments and the targets. So I wanted to give one example of how the approval of lecanemab in the United States impacted an ongoing clinical trial, the type of considerations that we've thought of. And I'll give the example of the AHEAD 3-4-5 study, which is a clinical trial in collaboration with the NIH and the Alzheimer's Clinical Trial Consortium in preclinical Alzheimer's disease. This trial is evaluating a 4-year treatment of lecanemab in individuals with intermediate or elevated amyloid in the brain who are cognitively unimpaired. And the intermediate amyloid is 20 to 40 centiloids. The elevated amyloid is approximately greater than 40 centiloids. And the dosing is based on the level of amyloid in the brain. So for instance, for intermediate amyloid, lecanemab is dosed monthly. For the elevated amyloid, lecanemab is dosed biweekly for a period and then transitions to a monthly maintenance phase. But we had to consider what would happen to people in the clinical trial if during this 4-year course of treatment they progress to early AD and therefore become eligible for an approved treatment. And whether we would just continue people in the clinical trial, whether they're on placebo or not, or as we ultimately decided, is that if people do transition to early AD during that period, they would then go on to open label lecanemab at the dose that's used for early AD. So in this case, we've adapted the clinical trial so that patients can kind of fail once they progress to early AD and then they move on to open label lecanemab. And of course, that has implications in terms of the statistical analysis and considerations in terms of how to manage that intercurrent event. But it's one example of how clinical trials are adapting. There's certainly a rationale for combination therapies in Alzheimer's disease. Alzheimer's is a multifactorial heterogeneous disease with a high unmet need. We know that the current anti-amyloid therapies, while they slow down progression, they certainly don't arrest progression. So there's opportunity for better benefit risk with combinations. There's a strong biological rationale for drugs targeting different aspects of the biology of Alzheimer's disease. There's preclinical data that can support some combinations. There's the opportunity, if there are additive or synergistic effects, that you may be able to reduce the dose of each component of the combination and thereby improve safety relative to a single drug alone. And then there may be the opportunity to treat later stages of disease if combination therapies target the biology that may be more active in that stage of disease. Certainly, regulatory authorities have expressed enthusiasm for innovative development approaches for delivering combination therapies. Physicians who treat Alzheimer's disease already use combination approaches with cholinesterase inhibitors and memantine. And in clinical trials, the anti-amyloid drugs were tested on top of these therapeutics as well. So yet another example of combination therapies that our physicians already have experience with. The biology of Alzheimer's disease provides a range of targets that can serve as rational approaches for combination therapies. So there's the amyloid pathway, which initiates early, the tau pathway, which initiates soon after amyloid and has a long early and late stage, eventually leading to a range of processes that lead to neurodegeneration. And these processes include synaptic dysfunction, neuronal injury and neuronal loss, inflammatory processes, vascular contributions, and then co-pathologies as well. So when we think of rational combinations, you can think of either two drugs that target different parts of the same pathway. Targeting production of amyloid and clearance of amyloid. The BACE inhibitors haven't been successful, but there are certainly gamma secretase modulators that are still under development. There's one can target amyloid and another pathology or pathophysiological process that's involved in Alzheimer's disease. So combining an amyloid therapeutic with a tau therapeutic, or an amyloid therapeutic and a neurodegeneration therapeutic, or combining treatments that can address co-pathologies in Alzheimer's disease, whether it's synuclein co-pathology, vascular co-pathology. There are non-clinical models that can be used to support combinations. Part of the challenges in these models is to accurately reflect the interactions between these different pathophysiological processes. So combining APP and tau transgenic mice can give mice that have both amyloid pathology and tau pathology. It's not completely clear that they're modeling exactly the process in Alzheimer's disease, where there's a long phase of existing amyloid pathology before tau spreads from the medial temporal cortex. Another example would be tau seeding models in APP transgenic mice. And then also taking advantage of the omics characterization of Alzheimer's disease or other types of modeling like QSP modeling to try to identify optimal parts of pathways to target and the particular timing. With the refinement of the staging of Alzheimer's disease and an understanding of the biomarkers in the different stages, this will also provide information as to when to initiate treatments, what pathologies are active in which stage of disease and which patient, and then target that particular population for the combination therapies that are being intended, and even the timing of the combination therapies. And you can see that the inflammation in particular can have different signatures over time in Alzheimer's disease. There appears to be early signatures during the preclinical phases and later signatures during the more developed and symptomatic phases of Alzheimer's disease. So understanding those signatures may help in terms of identifying at what stage of disease to test therapies that are targeting microglia and inflammation. The other key potential interaction between those therapies is recognizing that amyloid therapeutics and then some tau therapeutics function through microglial-mediated clearance. So it'll be important to understand the interaction of treatments that are targeting microglia and how they may impact the efficacy of the drug that's being used in combination, the anti-amyloid treatment. The biomarker strategy is also critical in terms of understanding the clinical development and the role of each drug in the combination. If we think about a biomarker approach in clinical development, we use biomarkers for patient selection, identifying the right disease stage, perhaps enabling precision medicine based on a person's biomarker profile, biomarkers demonstrating the effect of each drug in the combination in terms of target engagement and pharmacodynamic effects. But we really want to understand the impact of each drug individually. So is the effect additive or synergistic when the individual drugs are used in combination? And can that be detected by biomarkers and then ultimately by the clinical outcomes? And I wanted to give one example of how we've thought about combination therapy with an anti-amyloid treatment. And this is the DIAN-TU Next Generation platform in collaboration with Washington University. And this is a trial in autosomal dominant Alzheimer's disease. And it's testing an antibody against the microtubule binding region of tau versus placebo. And how we test it is dependent on the stage of disease in autosomal dominant Alzheimer's disease. For symptomatic patients, because they're symptomatic, they're in the early AD phase, we actually start everybody on lecanemab first for 6 months, and then randomize them to the tau therapeutic or placebo. With the idea that it's important for amyloid treatments to treat early in the symptomatic phases of disease. For the asymptomatic cohort, we actually start first with the tau therapeutic versus placebo for a year to try to identify the isolated effect of the tau therapeutic, and then we add open-label lecanemab after that. To look at what happens after adding lecanemab to that. So in a sense, using different designs in different stages of disease to try to identify the effect of each component of the combination. So in conclusion, it's a very exciting and innovative time in Alzheimer's disease, drug development, and clinical trial design. Clinical trial designs will have to take into consideration the evolving standard of care, the evolving understanding of biomarkers and how to define biomarker stages, thinking carefully about the selection or incorporation of amyloid therapeutics and the appropriate dosing regimen and approaches. And certainly there's a huge opportunity for combination drug development, given greater understanding of the pathophysiological pathways, the non-clinical models, the availability of regulatory guidance that supports drug development and combinations, and certainly taking into account all the advances that we're hearing about in this conference over this week. So thank you so much for the opportunity to present.
Thank you, Michael. It's a brave new world. Many, many questions. We now have another virtual presentation, but just to let you know, do please put questions in as well for the virtual presenters, because I believe they are joining us for the questions afterwards. So keep them coming, please. That would be brilliant. So the fourth presentation is Gary Romano from Alector, and he's going to be describing, in fact, he's combining really what we were just talking about in terms of clinical trials and new questions and inflammation. So he's going to be describing the phase 2 trial of a TREM2 monoclonal antibody in early Alzheimer's disease.
Good afternoon. I'm Gary Romano. On behalf of Alector and our partners at AbbVie, I want to thank you for this opportunity to share with you an update on the clinical development of AL002, a monoclonal antibody that activates TREM2 signaling and is under investigation for the treatment of early Alzheimer's disease. Alector presented phase 1 data and a phase 2 study design at the 2021 AAIC meeting. Today, I will discuss our progress in phase 2 and describe a clinical safety signal and resulting study modifications. These are our disclosures. Alector's scientific and therapeutic strategy is to harness the brain's innate immune system to treat degenerative brain disorders. This approach is not arbitrary, but is rooted in the genetics of neurodegenerative disease. Evidence from GWAS studies of people with sporadic Alzheimer's disease revealed that many of the loci that confer disease risk are involved in microglial function. These are illustrated in the figure on the right, indicated by the black arrows. The red circles are specific loci that are under study at Alector. Microglia are the primary innate immune cells of the CNS, and they play a number of important roles to maintain brain health and function, including clearing of misfolded proteins such as amyloid and other cellular debris, as well as maintenance of healthy synapses, astrocytes, oligodendrocytes, maintenance of the blood-brain barrier and the vasculature, and immune tolerance. Evidence suggests that with aging, there is senescence of the brain's innate immune system, which may impair microglial function, potentially contributing to the brain's vulnerability to age-related disease insults. Our hypothesis is that boosting microglial function may improve the brain's defenses against age-related neurodegenerative disorders. Triggering receptor expressed on myeloid cells, or TREM2, is a phospholipid receptor on the microglial membrane that senses pathological changes in the brain. Binding of TREM2 to its biological substrates, which include ApoE, lipids, A-beta, and other cellular debris, triggers microglial signaling pathways that allow the microglia to adapt a defensive response to disease by clearing pathology. Loss of function variants in TREM2 are known to be deleterious. Homozygous mutations cause an autosomal recessive disorder called Nasu-Arcola disease, which is characterized by a rapidly onset early dementia in early age with leukoencephalopathy and bone cysts. Heterozygote mutations in the TREM2 reduce the functionality of microglia and increase the risk of Alzheimer's disease. For example, the R47H variant increases the risk of Alzheimer's disease threefold. AL002 binds to TREM2 receptors, resulting in clustering of TREM2 in the microglial membrane and activation of TREM2 signaling pathways, which support microglial survival, proliferation, and function. In our phase 1 single ascending dose study, we demonstrated that treatment with single doses of AL002 resulted in dose-dependent reduction in CSF, dose-dependent target engagement, and microglial activation. On the left panel, you see dose-dependent reductions in soluble TREM2, which reflect AL002 binding and internalization of membrane TREM2. On the right, you see dose-dependent increases in CSF1R, a receptor tyrosine kinase that is essential for microglial development and survival. We have previously reported similar changes in SPP1 and IL1RN, which are also markers of microglial proliferation and survival. INVOKE-2 is our phase 2b study of AL002, which is now ongoing in patients with early Alzheimer's disease. This is a randomized, double-blind, placebo-controlled, common closed design study of up to 96 weeks of treatment with AL002 in 328 patients with early Alzheimer's disease. It includes 3 doses of AL002 that in phase 1 demonstrated target engagement and microglial activation. Participants received AL002 or placebo as monthly infusions. This trial was designed by Alector and AbbVie to be a biomarker-rich proof-of-concept study. As you can see here, the primary endpoint is the CDR sum of boxes. We've also included other clinical and functional outcome assessments. And biomarkers include CSF and plasma biomarkers for microglial activation and for Alzheimer's pathophysiology, as well as neuroimaging biomarkers, including tau PET, amyloid PET, and volumetric MRI. The study is nearly fully enrolled and remains blinded to treatment assignment. We expect to have last patient out in the third quarter of next year, with results shortly thereafter. Early in the trial, 3 participants presented with treatment-emergent neurological serious adverse events and associated MRI findings consisting of focal vasogenic edema, sulcal effusions, microhemorrhages, and superficial siderosis, resembling the features of amyloid-related imaging abnormalities, or ARIA, that have been described following treatment with anti-amyloid antibodies. Clinical presentations included focal signs, seizures, and mental status changes requiring hospitalization. These participants were treated with IV corticosteroids for presumptive treatment of ARIA, and all eventually recovered. Each of these participants that had SAEs were also found to be APOE4 homozygous. For convenience today in this presentation, I'm going to refer to the MRI changes we are observing in AL002 as ARIA-E and ARIA-H, although I'd like to emphasize that given the different mechanism of AL002, we do not yet know whether the biological mechanism causing these MRI changes are the same as for the ARIA that has been described as anti-amyloid antibodies. In the interest of patient safety and to continue the trial to learn whether AL002 provides benefit, Alector voluntarily made the following modifications to the original protocol. They implemented a dose titration that was gated by MRI surveillance at baseline, before each dose titration, and every 3 months thereafter. They implemented dosing guidelines for ARIA management. And given that at the time this occurred, all of the clinically serious cases that have been observed in APOE4 homozygotes, they excluded APOE4 homozygotes from further study participation. This trial has a independent data monitoring committee that reviews study data on a regular basis, as well as ad hoc for symptomatic ARIA cases. The IDMC agreed to these protocol changes and continues to recommend that the study proceed. In the interest of the safety of participants who may participate in other clinical trials of TREM2 or microglial modulation, we would like to take this opportunity to share with the research community details of the ARIA signal observed in our ongoing INVOKE-2 study. These are MRI images from 2 participants in INVOKE-2 who had radiographic changes resembling ARIA-E and ARIA-H. These are representative of most of the cases we have seen. On the left, images from a 77-year-old female in which MRI surveillance after dose 6 revealed vasogenic edema within the left occipital lobe, shown by the red arrows in the flare image. On follow-up exam 1 month later, the focus of ARIA-E was decreased in size and extent when compared to the previous exam. A later surveillance MRI 4 months later showed complete resolution of the previously noted ARIA-E changes. However, there was interval development of a single punctate focus of hemosiderin deposition in the area of the resolved ARIA-E, consistent with microhemorrhage or ARIA-H. On the right are images from an 81-year-old female who had onset of tinnitus 2 weeks following her first dose, and on MRI was found to have a small focus of sulcal hyperintensity, again shown here in the flare images with the red arrow at 16 days after dose 1. This small focus of sulcal hyperintensity in the right occipital lobe was compatible with ARIA-E. In her subsequent MRI, about 5 weeks later, the right occipital ARIA-E had increased in size, demonstrating both vasogenic edema and sulcal effusion. Concurrently, more than 10 incident microhemorrhages appeared in the right occipital lobe, indicative of ARIA-H. The next MRI 4 weeks later showed complete resolution of the ARIA-E. Microhemorrhages in the right occipital lobe, representing ARIA-H, remained stable. Now I'd like to turn to provide you with some data on the ARIA signal. starting with the temporal dynamics. In this study, most of the ARIA-E has occurred within the first 3 months of treatment and resolved within 4 months of detection. Here you see the mean and median for time to first occurrence of ARIA-E and time to ARIA-E resolution. Following discontinuation and exclusion of ApoE4 homozygotes from the study, we observed a marked reduction in the incidence of ARIA-E and ARIA-H, and also a reduction in the occurrence of severe ARIA-H. Here we're comparing incidence and severity of ARIA-E between the ApoE4 homozygotes who were enrolled early in the study and the current study population of non-ApoE4 homozygotes, which includes both ApoE4 heterozygote carriers and ApoE4 non-carriers. The top panel shows a significant reduction in the incidence of ARIA-E from 71% in ApoE4 homozygotes to 27% in the current study population. Radiographic severity of ARIA-E, which was modest in general, may be also slightly reduced in the current trial population. The bottom panel shows a similar marked reduction in the incidence of ARIA-H from 71% in the ApoE4 homozygotes to 29% in the current study population. Comparison of radiographic severity suggests a shift to less severe ARIA-H. I want to please note that this study is still blinded to treatment assignment and that these percentages were calculated conservatively, assuming that all ARIA occurs in the active treatment arms. The incidence of ARIA was higher in the ApoE4 carriers. In the top panel, you see for ARIA-E incidence that was lowest in the ApoE4 non-carriers at 19%, was slightly higher at 32% in the ApoE4 heterozygote carriers, and highest in ApoE4 homozygotes, 71%. The incidence of ARIA-H showed a very similar pattern, lowest in the ApoE4 non-carriers at 22%, followed by the ApoE4 heterozygote carriers at 35%, and highest in the ApoE4 homozygotes at 71%. In the current trial population, the majority of those with radiographic ARIA have been asymptomatic. Here you see that of 41 participants, or 27% of the current trial population that had ARIA-E, 88% were asymptomatic, 12% were symptomatic. None of the participants with isolated ARIA-H, that is, without concurrent ARIA-E, were symptomatic. The incidence of clinically serious ARIA, that is, those with SAEs, was just under 1% of the total participants dosed. In summary, treatment-emergent MRI findings resembling ARIA have occurred in a subset of participants in the INVOKE-2 trial of AL002, a monoclonal antibody that activates TREM2 signaling. These findings resemble the ARIA that has been reported following treatment with anti-amyloid antibodies regarding the MRI features, incidence, timing of onset, relatedness to the number of ApoE4 alleles, frequency and spectrum of associated clinical manifestations. Following the exclusion of the ApoE4 homozygotes from the study, the incidence and severity of ARIA was reduced. I want to emphasize that we do not yet know whether these events are related to amyloid clearance or other treatment-related changes in microglial function. We are collecting clinical and functional outcomes, as well as brain MRI, amyloid PET, tau PET, CSF and plasma fluid biomarkers in this study, and expect to report those data late next year. I want to thank the INVOKE-2 investigators and especially the participants in this trial for helping us to understand the safety and potential benefits of this mechanism for the treatment of Alzheimer's disease. Thank you for your attention.
Thank you, Gary. Can I just check, do we have the two virtual presenters able to, we do, to answer questions. Brilliant. Thank you. Okay, we've got 10 minutes for questions, and there's lots that's come in. Please keep them coming in. That would be great. I have a question, first of all, for Dr. Kazaglu. At what stages and in which types of AD would you predict your antibody to be most effective? That's the first question. And then, are you also planning, which was also my question, are you also planning to test it in other conditions? And if so, what?
Yes. So over 80% of Alzheimer's patients have vascular pathology. It could manifest with microbleeds, white matter hyperintensities, leaks of the blood-brain barrier. So the fibrin is present across multiple patients in AD. And of course, given the biomarker data about its presence very early in mild cognitive impairment, I think early stages would make a lot of sense to treat, but the treatment, of course, can be used throughout the course of AD. Regarding other conditions, obviously, fibrin is not present only in Alzheimer's disease, but across the neurodegeneration space. And this is ranging from traumatic brain injury to COVID-19 neuropathology to postoperative cognitive decline and delirium, Parkinson's disease, epilepsy are all cases where this pathway could be active by neurotoxic polarization of microglia cells. And we hope that the fibrin immunotherapy could be in the toolbox of clinical toolbox to neutralize blood toxicity across neurological diseases with blood-brain barrier disruption of autoimmune origin, trauma, and neurodegeneration, including also progressive multiple sclerosis, which is an area that we have a lot of preclinical data.
Thank you very much for that answer. We also have people in the room. Please do come up to the microphones if you'd like to ask a question. I've got two patiently waiting. So microphone number 2, please state your name, where you're from, and who the question's for.
Sure. Nilüfer Erdoğan-Tainer, Mayo Clinic. The question is for the second and fourth speakers. It is clear from human genetics and multi-omics studies as well as model system studies that certain innate immunity genes, if overexpressed or if their function is enhanced, it's beneficial for Alzheimer's disease and related outcomes, and yet for TREM2, PLC gamma 2 being examples, for others it's detrimental. So in the context of this microglial molecular complexity, how should the field be thinking about developing, prioritizing, and eventually applying therapies in the pathway of innate immunity?
That's a great question. Michael Henniker, do you want to take that first?
Yes, thank you. That's a great question, and I think it's one of the key questions for modulating microglia and other immune responses in the brain.
Recording in progress.
The point is that I believe we should have more biomarker information with respect to inflammatory signals. We need imaging biomarkers, we need blood and CSF biomarkers to address this. By now, we have a lot of biomarkers which assess neurodegeneration, amyloid beta deposition, and we have to make the same progress in the field of innate immunity. One of the subsequent speakers showed this nice model with the two waves of inflammation, which may be actually more complex than just being two. Inflammation is something which works to resolve and to heal, but the chronicity which sets in and the dynamics of amyloid beta deposition as being one of the major danger-associated molecular patterns gives it that there might be multiple waves of different natures in the same brain. So I think we have to have a greater insight in the detail of the inflammatory cascades which are present at a given time point in the patient brain.
Gary, do you want to add anything to that?
Yeah, thanks. I agree with everything Dr. Henniker had to say. It's an excellent question. It's a key question. And certainly you're right to point out that it's not so simple with regard to microglial function. And there's certainly evidence from preclinical studies suggesting that, on one hand, microglia may help contain A beta plaques and delay disease progression, and other data suggests that microglial activation may contribute to tauopathy, neurodegeneration. You know, while these preclinical models are certainly informative about the biology and very helpful, they can only take us so far in understanding whether microglial-based therapeutic strategies are going to be effective in patients. So our philosophy is we have to start somewhere with clinical studies. if we're going to learn whether and how and when in human disease they may provide benefit. In our case, we're following the genetics that tell us that TREM2 loss of function variants increase the risk of AD, and testing a hypothesis that boosting TREM2 signaling will slow disease progression. That may be a little simplistic, but it's a good place to start, we think.
Thanks.
Thank you very much. I'm going to go to microphone 1 now, and then my co-chair wants a question.
Hello, my name is Bart Stroper. A question for Dr. Irizarry. Regarding the DIAN trial, I am very confused about the protocol where you first use the tau antibody and then the amyloid therapy in preclinical phase. So I would expect, especially in the DIAN patients, which are presenilin and APP mutations, that you have first amyloid pathology and then the other pathologies. And so and the second aspect is that but that's an hypothesis, maybe you can address that too, that antibody will only work if you have amyloid plaque, so not to prevent amyloid plaque, so because of the working mechanism, microglia, etc. So I don't understand very well the rationale for the DIAN trial, if you could explain that.
Certainly. So the focus of the DIAN trial is to test the tau antibody. And we still, But
wouldn't you test that once the pathology is there? Because
so the preclinical, we did want to test it in the preclinical stage where people did not have symptoms, did not yet have really signal on tau PET, but where they're in the early tau stages, actually. So we did want to test it independently first in the preclinical stage in people where they might have elevated phospho-tau already, but not yet tau.
But they have already amyloid plaques.
They have amyloids, but we wanted to see the independent effect of the tau antibody. And then after a year, everybody goes on the amyloid antibody as well. So they still get the amyloid antibody. But because we felt that in the symptomatic people, it was important to give the amyloid antibody first for 6 months and then add the tau. So because it's a small patient population, we set it up that way in order to be able to identify an independent effect of the tau antibody. But we are targeting, you're right, in the preclinical stages, first just the tau antibody alone in people that have elevated amyloid, but probably no tangles yet.
And do you think that lecanemab would work in these patients before amyloid plaques are there, or are there always amyloid?
So roughly all these patients have amyloid already, yeah.
Can I ask a quick follow-up on that? Mm-hmm. So we know you get downstream effects with treating with the anti-amyloid therapies, including changes on tau and NFL. So how are you going to disentangle those downstream effects from the direct effects of the anti-tau therapy?
Because everybody will be on the same backbone of anti-amyloid therapy for the same period of time, we'll be looking at the comparison of people that are added the tau antibody on top of that versus placebo. Mm-hmm. So we'll be able to do that comparison, but it may be on a background where there is some dynamic changes in these biomarkers. We're also looking at biomarkers that may be more directly related to the tangle pathway, like tau 243, versus the biomarkers that are maybe reflective of amyloid, like the phosphorylated tau species. But it's an ongoing, very evolving area.
It's complicated.
I have a question to Gary. First, I'd like to congratulate you for focus on showing the adverse events so early in the trial. This is very nice and very important to inform the other trialists testing the same target. But my question is, when you talk about ARIA, is a construct that you have in the brain edema or bleeding together with amyloid removal. Do you have any data that you did not show that you are linking these side effects with the amyloid removal? You have this data, or is something that you imagine that is the case?
No, thanks for asking. No, we are collecting data. We will have amyloid PET and a number of other fluid biomarkers that we think will address this, but we have not. We are not looking to that data yet. We've agreed with our partners at AbbVie to maintain the integrity of the trial and open that data at the end of the study. So, as I said, we don't at this time know that this signal is related to amyloid removal.
Okay, I have some online questions. I think we've got room for maybe one or two more questions before we have to stop. So a couple of people are asking, and this is a question for Michael Henniker. And the question is, would it be possible that microglia also phagocytose reactive astrocytes, and would that be a biological interpretation for increased GFAP levels in the blood and other reactions?
And that's a very interesting question. And as far as I would say that the only evidence I know from for phagocytic activity has so far been in vitro. I would love to see that in a 2 or 3 photon LSM experiment in vivo and learn more about the conditions, how microglia cells take on astrocytes.
Thank you. And one for you, Michael. How would you combine your anti-amyloid treatment with a microglial treatment? You gave a very interesting change in dynamics of inflammation within the course of disease. So what would be your best guess?
I think first I would see whether I could assess in a non-clinical model whether the mechanism of action of microglial-mediated clearance of amyloid or protofibrils is impacted by targeting microglia. So first to see whether the combination would actually interfere with an amyloid therapeutic. And then I think it will depend on the signatures of the microglia in the various stages of disease, which I agree are still being elucidated to try to identify when it might be optimal. And if there is interference with the way an anti-amyloid drug works, you may want to try something sequential or focus in different stages of disease.
Thank you. We've run out of time, which is a shame because we've got loads more questions. So those people that have asked questions, we'll try and get answers for you offline. But thank you very much, and please thank all our speakers for some terrific talks.